Hello, in this video we will discuss about the bacteria. So this is the bacteriology lesson. So let's begin to understand first of all the general structure of the bacteria. So this is the general structure which that is showing the flagella, pili as well as cytoplasm and cell wall. So the bacteria is a prokaryote, unicellular microscopic microorganism. So this is we can understand from the microscope because it is the minute microscopic a living organism. It is living basically. So let's begin to understand one by one to understand their different part of the bacteria. So first of all, the flagella. Flagella is can be three, four, or some. So the different type which that is used for motility, organ of locomotion, and or hair-like structure and cytoplasmic appendages. The flagella. So this is the different type on the basis of the flagella. The bacteria is different type. For example, Lophotrichus, Peritrichus, and Atrichus and Monotrichus. So you can see that is the flagella arrangement. As well as Peritrichus is basically is the whole. Uh, uh, Peritrichus mean it structure is basically the whole body is covered with a flagella. So here is the cytoplasm. Cytoplasm means the inside the cell membrane. The cytosol is for normal living organism. And homeostasis. So in this way, the cytoplasm is possible due to the living organism. So without cytoplasm, no life will be possible of the cell. So the cell membrane is the bilayer of phospholipid which that is covered with the cell wall, and the cell membrane is the bilayer of phospholipid which that is used for the barrier for the semi-permeability of water absorption, etc., etc. So on the other hand, here is the you can see that is the capsule, is the outer most layer of the bacteria which that is outermost from the cell wall. So this is the sticky substance that form protective layers called capsule, and outside the cell wall contain polysaccharide which a few capsule peptidoglycan provide resistance against the antibiotics. So this is used for against the pathogen or against the antibiotics which that will kill the bacteria, so it is the resistance barrier. So this is play important role as an immune system of the bacteria. So the pili or fimbri are present, which that is shorter than the flagella, and it is the contain protein pillin. There is used for cell to cell attachment for sexual reproduction, as well as it is used for the attachment in the surface also. So it is the shorter and made up of pillin, while the flagella is made up of flagellin protein. So this is the F pillus. Let's begin to understand the endosome. So this endo mean inside, endospore formation or endosome. It can be exosome or endosome, but it is the endosome. So this mass of protoplast surrounded by multi-layered wall, outer wall exosporium, inside the multi-layer of protein, thick coat formed of peptidoglycan, and it is made up of inside deposition of calcium and uh, uh, and dipicolic dipicolinic acid which that is used for the heat resistance. While here is the red color is showing that the plasmid, this is the extra DNA material. This is used for the vector, and it is also known as vector. And here is the nucleoid. This is the chromosomal DNA. It, in this way, the bacteria contain a one chromosome in the form of circular DNA. So it is the nucleoid is the circular and is used for protein synthesis and control the genetic activity of that bacteria, while the plasmid is also, and plasmid is the extracellular DNA. It is used for transformation during the sexual reproduction to the other cell and antibiotic resistance due to the production of protein which that is used for the antibiotic as well as recombinant bacteria and formation. So the recombination will be occur due to. On the other hand, the gene of interest can be inserted during the biotechnological aspect. It is also used. And here is the spores or cyst. Cyst means that it is bind with the membrane side. So you can see that is a bacterial resist by spores either within cell. So the endo or external to cell, exo, can resist the heat, desiccation and radiation during the harmful environment. It will be dormant state of the bacteria, which that will again produce the bacteria during the favorable condition. Here is a blue color is showing that the gas vacuole. This some aquatic organism contain gas vacuole, provide buoyancy. It is impermeable to water. So it is impermeable to water. That is why the bacteria can float on the surface of the water, and that means the buoyancy will increase the surface gliding or something. So here is a ribosome. Ribosome in the form of smaller subunit and larger subunit to become a 70S. And this is the ribosome is used for the synthesis of protein from the messenger RNA. So let's begin to understand the microscopic study, which that is showing the different slide to understand the structure of the DNA, structure of the bacteria on the basis of their shape and their structure. So let's begin to understand the shape. On the basis of the shape, we can give the name. So the bacillus, if we will discuss about the capsular-like structure, is looking like this, while the diplobacillus means that two cells combined together, and staphylococcus, it is here, which that is staphylococcus, because the cocci will group together. And streptococcus. Streptococcus means the thread-like structure which that is made up of cocci. And on the other hand, the streptobacillus in the form of thread-like structure made up of bacilli bacteria. And here is the palisade-like structure is looking like this. And here is a vibrio. It is the coma-like structure. While the tetracoccus is made up of four number of cocci, so that is the tetracoccus. Our tetrabacillus is also made up of four. So the cocci is the bead-like structure, while the diplococcus is the two bead containing. And here is the spirillum, which that is a spiral-shaped bacteria, which contain a flagella also. So let's begin to understand on the basis of gram-positive cell wall and gram-negative cell wall. So how it's possible, let's begin to understand. Here is the cell of the bacteria, which that is showing the membrane of the cell. So you can understand as well. So here is the two gram-positive cell wall and gram-negative cell wall, which that contain a different content we will discuss now. So here is the teichoic acid are present the blue color, and here is the orange color showing that lipoteichoic acid, and here is the periplasmic space, and this is inside is the plasma membrane, and integral protein are present in yellow color showing in the figure. So the example of these a gram-positive bacteria is the cocci and bacillus, while the next is the gram-negative bacteria, which that is on the basis of we are distinguish between the gram-positive and gram-negative cell membrane. So here is the upper part is known as a specific side chain, and inside is the porine. This porine used for the movement of ions or something. And here is the lipopolysaccharides layer are present, and inside is the phospholipid, and the brownest lipoprotein are present is here, which that is bind together with the peptidoglycan, pink color showing in the figure. So the example is the cocobacillus and bacilli and cocci. That's basically is the example. So let's begin to understand the reproduction, the growth and reproduction in bacteria. So the growth I am not showing here, but you can understand the growth is the lag phase, lag phase, stationary phase, and death and decline phase on the passage of time. So the increase of the number of cells in 20 minutes required for the cell division. So that is the asexual reproduction. We will not understand about the growth, but here I am not drawing the graph, but next is the method of recombination. The sexual reproduction we will discuss first, and next we will discuss about the asexual also. But this is the sexual, because here is the plasmid, the DNA polymerase and replicase enzyme used for the chromosome with F negative factor bacteria is contained because it's want to donate the genetic material. So the genetic material used for the synthesis of protein for antibiotics, and this is the plasmid, for example. And here is the donor and recipient. Recipient is known as F negative, while the donor is known as the F positive. So the F positive will donate the after the replication of DNA, the plasmid will donate in by the sex pillars is the conjugation. So this is the conjugation, and this is the conjugation tube formation will be occur. And in this way here is the plasmid will synthesis the protein from messenger RNA and the polysome formation due to the number of ribosome will be attached with the messenger RNA to synthesis protein, and this protein will increase the surface protein. This surface protein can be different for resistance against the antibiotic and other things. While here is the transformation is another mechanism. In this way, the transfer without conjugation. So the complete plasmid will be transformed into the empty uh, empty cell. So you can see that is if the dead uh, pathogenic bacteria so will contact with the non-pathogenic bacteria, so the transformation will be occur to synthesis the protein which that become a pathogenic bacteria due to the synthesis of protein. While on the other hand, here is the gene of interest during the biotechnological field, which that is provided by the recombinant plasmid to provide into the cell of the bacteria. After the bacteria, which that is introduction with the recombinant vector used as a plasmid used as a vector, so which that is provide and increase the protein, for example the insulin production due to the recombinant plasmid after the insertion of the DNA segment of insulin producing DNA segment. So the new protein for antibiotic resistance or etc., which that is produced. On the other hand, important thing is that the biotechnological field will produce the different type of desired substance due to the gene of interest insert into the vector to insert into the bacteria. So that here is another topic is the third party, that is the transduction. Transduction means the uh, bacteriophage which that is used for the insertion of the DNA from the bacteriophage after the injection, and that bacteriophage will bind the DNA of the bacteriophage will bind with the chromosome of the bacteria, and after this binding that's it, the two cycle will begin, the specialized transduction and generalized transduction. Specialized transduction is the lysogenic cycle, while the generalized transduction is a lytic cycle. It means the lytic cycle means the lysis of the bacteria will produce more bacteriophage, while the lysogenic means the gene will transfer after the replication of the DNA. So the reproduction asexually is the binary fission and endospore formation, while the sexual reproduction is the bacterial recombination and transduction and transformation. So let's begin to understand about on the basis of nutrition in bacteria. So the bacteria consume energy differently. Autotrophic bacteria. First of all, we will understand about that. Autotrophic will use the carbon dioxide, and this is the use the carbon dioxide and Heterotrophic is another topic we will not understand. So the heterotrophic is another, but autotrophic is the another. Heterotrophic you consume as a decomposer, while the autotrophic is the photoautotroph, purple sulfur autotroph, green sulfur, and purple non-sulfur containing, which that use the carbon dioxide to produce energy. While the chemoautotroph is the chemical with oxygen to use. So the nitrifying bacteria, iron and sulfur, and hydrogen bacteria, which that is nitrosomonas is important. So that was the important things about this. While the growth phase, we will not understand about the growth of the bacteria, but next video we will discuss. So thanks for watching.